What is AnVIL? The main objectives of this tutorial are to highlight the revolutions in DNA sequencing and computational genomics that are pushing researchers to compute where the data resides, introduce the main components of AnVIL, and understand the roadmap for data, infrastructure, and events. Goals of AnVIL. 20 years ago, Sanger sequencing enabled our first complete glimpse into the 3 billion letters that make up the human genome. As these instruments could only produce up to 1,000 nucleotides at a time, entire warehouses were filled with these machines. The 2010s introduced breakthroughs in massively parallel DNA sequencing. While this technology initially only produced 25 nucleotides at a time, these machines were able to complete billions of reactions in a few days, and importantly, at orders of magnitude less cost. These innovations not only enabled researchers to sequence whole genomes faster, they inspired the creation of assays to measure gene expression, chromatin structure, DNA modifications, and more. After 10 years of steady improvement, today's workhorses such as the Illumina NovaSeq enable genome sequencing of entire populations in not decades, but years if not months. With so much sequencing data being produced, the bottleneck has shifted from data production to data analysis. Fortunately, similar changes have also occurred in the realm of computational genomics. When the first human genomes were coming out, virtually all computational work occurred on institutional compute clusters. These clusters were hard to scale and expensive to support. The 2010s saw the first cloud-based applications that promised the ability to speed up analysis by spreading it out across hundreds or thousands of compute servers on the Internet. While many proof-of-principle tools were created, the management of cloud-based resources remained technically challenging and out of the reach of most researchers. Today, platforms like AnVIL work to revolutionize computational genomics by creating highly scalable cloud environments designed with proper data security and interoperability between major cloud resources. Importantly, AnVIL aims to be the place for not only computational experts but for everyone that desires to conduct computational genomic research. The goal of AnVIL is to invert the model of genomic data science. Rather than bringing data to every researcher, AnVIL brings the researcher to the data by providing secure access to data, cost-efficient scalable compute resources, and a collaborative analysis environment where researchers can share and publish computational research. What is AnVIL? Three main features of AnVIL are: a cloud-based infrastructure that is cost-efficient and scalable, secure data access that conforms to regulations, and a collaborative computing environment where researchers can share and publish computational research. The three main components of AnVIL are data, workflows, and compute. The Gen 3 Data Commons brings data to the forefront by allowing you to search for and assemble cohorts of data relevant to your research question. DocStore provides an open platform where researchers can share cutting-edge analysis tools as well as complete reproducible workflows. Finally, Terra provides the backbone for the analysis by orchestrating all the necessary cloud resources for storing data and running computational analyses. AnVIL allows many modes of interaction to conduct analysis. Core to Terra is the ability to run workflows that can scale to hundreds or thousands of different compute nodes. Interactive analyses are also possible through both Jupyter Notebooks and RStudio, both of which are configured to run the complete catalog of packages available in Bioconductor. Finally, the web-based Galaxy platform will soon be available, providing access to an international community of reproducible workflows and open-source training material. An important note is that AnVIL provides a FISMA moderate environment. This satisfies the security requirements for many controlled access datasets. Let us now briefly introduce the main components of AnVIL. First is Terra, which allows for highly scalable analysis workflows. This mode of batch analysis parallels the kind of work typically done on a high-performance compute cluster. Workflows are implemented in the workflow description language, or WDL for short. We will discuss this more in depth in the interactive analysis course. Interactive analysis can occur through the Jupyter Notebook interface. Most often used with Python, Jupyter Notebooks allow for transparent code, visualizations, and narratives. Interactive analysis can also occur through the RStudio interface. Importantly, this R-based environment provides familiar access to users of the Bioconductor ecosystem. The latest release of Bioconductor offers almost 2,000 software packages to analyze high-throughput genomic data. Accessing data on AnVIL current requires users to search through Terra workspaces or identify datasets through another means. An interface between Gen 3 and AnVIL continues to be developed and when ready will allow users to more easily search through hundreds of thousands of datasets to create virtual cohorts based on clinical attributes of interest. Once Gen 3 is fully integrated into AnVIL, Gen 3 will dramatically enhance data search. Shown here is an example of the Gen 3 Data Explorer where researchers can search and design patient cohorts on the fly from existing samples. DocStore is a registry of tools and workflows. Users and organizations can publish workflows that can easily be imported into AnVIL. These workflows are created using the WDL workflow language. Coming soon is the web-based Galaxy platform for running analysis. Galaxy brings a community-contributed toolshed of almost 8,000 tools that can be easily assembled into workflows without any programming knowledge. Integration into AnVIL is expected by October 2020. If you don't see an analysis mode that you need, don't despair. Not only can you bring your own tools and workflows, you can leverage a powerful AnVIL API which allows you to build your own cloud-based applications using AnVIL components. These APIs allow you to benefit from all the engineering work to create a secure, scalable platform. What's next? AnVIL currently provides access to GTX, 1000 genomes, CMG, cCDG, and eMERGE datasets. Many datasets are being made available including COVID data. Here is our infrastructure and tools roadmap. Finally, join us for a training event or collaborative research festival. In this tutorial, we presented the breakthroughs enabling a paradigm shift where researchers are brought to the data, introduced the main technical components of AnVIL, and discussed a roadmap for data, infrastructure, and events. Next, we will discuss important concepts in cloud computing, cost considerations, and several use cases. Thank you to all the people that helped.